Assalamu alaikum wa rahmatullahi wa barakatuh. Hi, ladies and gentlemen. Really nice to see everybody here. We're sorry for the late starting of this session, but I hope, as they say, all good things come to those people that wait. So I'm not going to talk much. I just want to introduce our first speaker of this session. It's Dr. Rachel Tanner. Rachel is an associate professor in One Health at the University of Oxford, where she completed her undergraduate degree in biological sciences and a DPhil in clinical medicine. I've known Rachel a long time. She's an excellent scientist. She's done her postdoctoral fellowship, and now she started her own group. Her research is concerned with tuberculosis in humans and cattle, with a focus on vaccine design and evaluation. And in particular, she's interested in immune mechanisms and correlates of protection, characterizing the humoral response to TB vaccines and novel antigen discovery. So I'm going to stop there and hand the floor over to you, Rachel. Thank you very much.
Thank you very much, and special thanks to Ahmed for inviting me and giving me the opportunity to speak, and to the organizers and for everyone for being here today. So I'm going to give the first of two talks about TB and give you an overview of some of the latest advances in TB vaccine development, and then towards the end show a little bit of some of the new data coming out of our lab on antigen discovery. So I'm sure everyone's aware of the current status of the TB epidemic, so it's still a major global health problem. So 2021 figures from the WHO showing that 10.6 million people get ill with TB or develop TB disease every year. And the COVID pandemic's really set back progress that was being made in the fight against TB. It's also, again, the leading cause of death due to infectious disease. So temporarily taken over by COVID, but now back to the dubious top spot of being the leading infectious disease killer, so 1.6 million deaths a year. And TB is really a One Health problem, so it not only affects humans, but also animals, and it's particularly a problem in cattle, so in both LMICs and the UK, it commonly affects cattle, so we slaughter about 50,000 cattle a year as a result of bovine TB. And there's transmission both from cattle to humans and more rarely from humans to cattle, but it also infects wildlife, and that means that you get wildlife reservoirs, and it can also reside in the environment. So there's really an interaction between those three components that needs to be taken into account and looked at holistically in an interdisciplinary way in order to really tackle the problem. So we all know that prevention's better than cure, that vaccines are generally thought to be the most effective strategy for controlling epidemic and pandemic diseases. We do have a vaccine for TB, which is the BCG vaccine. It was first licensed over 100 years ago, and it's still the only licensed vaccine after all that time. But the efficacy that BCG confers is extremely variable. So although it gives good efficacy in the UK, it's around 80%, the efficacy in the closer you get to the equator wanes. You get even negative efficacy in some studies, for example, in India. Similarly, it confers only incomplete protection in cattle. So you get around 50% of cattle showing signs of protection following BCG vaccination. And actually in the UK, the use of BCG is prohibited because the control strategy is test and cull. So there's constant testing essentially, and surveillance of cattle using the tuberculin skin test. And because that BCG sensitizes cattle to a positive tuberculin skin test, it's not possible to use it in combination with that test and cull strategy. This is just highlighting the variable efficacy across different populations in humans. So development of a new vaccine is really a priority to end TB, both in humans. This has been recognized by the WHO, saying that in order to reach the end TB strategy targets, a new vaccine will be required, and it needs to be effective across all age groups. And similarly in cattle, this was a response to a big government review of a government developed strategy. I can't think of the word, sorry. But they reviewed a study and showed that one of the main priorities is acceleration of work to develop a vaccine against bovine TB. So this is the TB vaccine pipeline in the year 2000. You can see this is purposely left empty because there was very little to nothing going on at that time. But you'll be pleased to know that by now, 2022, this is the latest update in October 2022, that there's actually quite a lot happening at various different phases, from preclinical right through to phase 3 clinical studies, and various different approaches, some of which I'm going to go on to highlight now. So I'm going to structure this around various different approaches and then give some examples of the leading candidates within each approach. So one of the first possibilities is to keep using BCG, but to try and improve on it in some way. So there's a few ways to go about this. One is to boost, so to have a BCG prime and then a homologous boost, BCG revaccination. Another is to use BCG, but administered by different routes. So I'm going to talk to you a bit about intravenous BCG and also BCG by the aerosol route. And another is to genetically modify BCG to make a recombinant strain that's more effective. So starting with BCG revaccination. So this idea was first tested in a few studies in Brazil in the 1990s, 1980s, 90s, and it didn't appear to have much effect. And so people really didn't pursue the idea much further than that. But actually, recently it's had renewed interest following this study in South Africa. So this was really designed to be an efficacy study of H4IC31, which is another TB vaccine candidate, but they also included a BCG revaccination arm on that study. So these are healthy adolescents who are HIV and MTB uninfected. They had received BCG vaccination routinely at birth, and then they were randomized to either receive BCG revaccination, this new candidate, H4IC31, or to be unvaccinated placebo controls. And the primary endpoint was quantiferon conversion, so MTB infection, with a secondary endpoint of sustained conversion, because people can convert and then revert, but we're looking in this case against protection against sustained infection. These are the results, and so you can see that by the primary endpoint, the H4IC31 in the last column, it had relatively low efficacy against quantiferon conversion and almost significant but not quite significant efficacy against sustained conversion. BCG revaccination, perhaps surprisingly, actually had significant efficacy against sustained quantiferon conversion. And this caused quite a lot of excitement in the field. And using samples from that study, people are doing a lot of work now to understand biomarkers of protection from sustained infection using those samples. And there's also a validation study of this trial underway where they're looking at 1,800 quantiferon negative adolescents, a much bigger study to see if this is really a true effect. So the second approach to improving BCG is to use it by different routes. There was a large study in Nature a couple of years ago from Bob Cedars' group in the NIH in the US, where they showed that you could prevent TB in macaques after administering BCG by the intravenous route. So this is unlikely to be a deployable strategy in human infants, but it's an unprecedented level of protection that hasn't been seen before with a TB vaccine. And that's a really critical situation in order for us to understand correlates of protection. An excellent example, exemplar for us to understand correlates of protection. So this is some of the data from that study. So each of the groups are different routes of BCG vaccination, and in each graph is a different measure of pathology following MTB challenge that was given after the vaccination. And so IV is the group in the middle in red, and you can see that by all of those measures, it has much lower pathology than any of the other routes. And in fact, by most measures, most of the animals have no pathology at all, so they're actually sterilely protected, which really has never been seen before. This is a CT scans of their lungs. So in this figure, the rows are different routes of vaccination. The fourth row down is the intravenous. And you can probably notice that there's really not much pathology, if any, going on in the lungs of those animals. So this intravenous BCG really prevented infection from establishing. The other option is to give BCG by an aerosol route, so delivered to the site of natural infection. And Iman's going to talk about this a lot more in the next talk, so I won't talk much about it, but just to note that this is not a new idea. So this is a paper from the 1960s saying that aerosol vaccination is as old as the disease itself. But there is renewed interest, and with new technologies, it's taking off again as an area to pursue. So the third approach to improving BCG is to use these genetically modified recombinant strains. And I'm just going to note one of the leading candidates here, which is VPM1002. This is a BCG that's engineered to express Listeria lysin, and that means that it can perforate the phagosomal membrane, and therefore you get more release of antigen into the cell, more antigen presentation and cross-priming of dendritic cells, and therefore more activation of T cells. It's recently completed a phase 2 trial. This is double-blind in over 400 South African neonates, and they either received BCG or VPM1002. And VPM1002 was non-inferior in terms of safety. But if you look at the levels of immunogenicity, so interferon gamma following re-stimulation, levels of polyfunctional T cells, they're actually lower at most time points following VPM1002 than BCG. But a phase 3 efficacy trial looking for prevention of disease is currently underway, and it's expected to complete late this year, early next year. Moving on to BCG replacements, one of the leading candidates is using an attenuated strain of MTB itself, which is called MTB-VAC. And this is the rationale behind the design of MTB-VAC. So if you think about the way that BCG was developed by sequential passage in order to attenuate it, now you can actually target regions in order to attenuate. And this is Carlos Martin's group in Spain. So the way they went about this was to look at an M. bovis infection that had been particularly virulent in a human population in the 1990s. And then they tried to understand what it was about that strain that made it so virulent. And they identified this FOP region in particular, which made it had high expression in this strain and appeared to confer that virulence. And they then got a MTB strain and knocked out that FOP region to make it attenuated. So it's gone through a lot of preclinical and clinical development. So starting in 1997, lots of preclinical testing in mice and guinea pigs and non-human primates, showing that it's safe and immunogenic and effective, and it's now in phase 2A and phase 3 trials. This is just a snapshot of the efficacy data from non-human primates. So MTB-VAC is on the right of each graph, and again, you're looking at levels of pathology. So you can see it has significantly lower pathology than BCG by most measures. So moving on to the third approach to vaccine development, this is to boost BCG with a subunit vaccine. So rather than your homologous prime boost where you're giving BCG followed by another BCG, here you would prime with BCG and then give a particular antigen by varying strategies in order to boost that response. And the main ways in which you can do this is through a recombinant viral vector, for which the Jenner is probably best known, or to give a protein vaccine in combination with an adjuvant. So looking at these viral vector vaccines, I'll talk a little bit about the ones coming out of Oxford over the last decade, which is predominantly those expressing the antigen 85A. So this was a large phase 2B efficacy trial led by Oxford in South Africa, which recruited almost 5,000 infants. And then they were randomly allocated to either receive a placebo or to receive MVA-85A. And all were BCG vaccinated at birth, and they were followed up for 3 years. So this was the efficacy data. It's obviously quite disappointing to see that there was no significant efficacy. So the primary endpoint was development of TB disease, and there was no difference between the placebo controls and the infants receiving MVA-85A in terms of the number of cases of TB disease. So what's next for MVA-85A? Well, there's been a couple of trials since looking at it, for example, by aerosol delivery. And this shows that if you look in the bowel, so in the lung, the bronchoalveolar lavage fluid, T-cell responses specific to antigen 85A are stronger when the vaccine is administered by the aerosol route rather than intradermally. And so it might be that by inducing stronger immune responses at the site of initial infection, you can actually confer superior protection. Obviously, further studies are required. And then this is looking at 85A in the ChAdOx vector, which you've probably heard about. And this is a prime of ChAdOx 85A boosted with MVA 85A. So the final approach is to use a protein with an adjuvant, and arguably the leading candidate in this setting is the M72 AS01. This is a GSK candidate which consists of a fusion protein of two MTB antigens combined with AS01. And it has been shown to elicit good proliferation of T cells and interferon gamma production in people that have latent TB infection or TB disease, and to have an acceptable safety profile. So recently, a phase 2b control trial results have been released. This was in African adults who were MTB infected, so they were looking at development of TB disease or prevention of TB disease. And so this is the immunogenicity results. So there was good induction of antibodies, good induction of polyfunctional CD4 T cells. And then looking at the efficacy, it was quite promising. So an overall efficacy at 3 years of 50%, which in the TB world is it generated quite a lot of excitement, and actually that's now gone into a phase 3 prevention of disease trial with a much larger number of volunteers. So for the last part of the talk, I'm going to talk more about what our lab is working on at the moment, which is the role of the antibody response. So traditionally, TB research and vaccine development has focused almost solely on the cellular response because MTB is intracellular, and so it makes sense. But actually, there's a growing body of evidence that antibodies may play more of a role in protection than we previously understood. So in particular, these two papers, this Liu et al. Cell paper from 2016, showing that antibodies from individuals with latent TB infection, who are considered to have some degree of protection, have different functional patterns and glycosylation patterns than those taken from individuals with active TB disease. And then also Li et al. In 2017 showed that healthcare workers who are highly exposed to TB, so they have a lot of exposure through work, but remain uninfected. If you take antibodies from those individuals and passively transfer them into mice and then infect the mice with TB, the mice show some level of protection. So it seems like antibodies are doing more than we thought. As I mentioned before, we know that BCG vaccination is partially protective in some species and populations and by some routes. And so the rationale of this body of work is that we can learn from BCG in situations where it is protective. We know that antibodies are induced by BCG. Do they have a role in protection? There is some evidence in vitro, and also this Fletcher et al. Study in 2016 came out of our group in Oxford looking for correlates of risk of TB disease, found that antigen 85A specific IgG was associated with reduced risk of reduced risk of disease. What we don't really know is the mechanisms of antibody-associated protection, and so that's something that our group's working on, looking in functional assays. And we also don't know the target antigens of BCG-induced antibodies. So that part's what I'm going to talk a bit about now. So the way we went about trying to identify these targets is through a whole proteome microarray approach. So this was in collaboration with Antigen Discovery Inc. in California, and they have arrays spanning almost the entire proteome of MTB, so over 4,000 proteins. And obviously this method is very high throughput and unbiased, and it's essentially a very large ELISA, and so proteins are printed onto each well, and then you add your serum and you add a secondary antibody, and then that has fluorochrome, which allows you to quantify the amount of protein reactivity in that serum sample. So this was a design of our pilot study. The sample sizes are quite small because this is very expensive, but we looked at 10 non-human primates who received BCG by either the intradermal or the intravenous route, 10 humans who received ID BCG, and then also some cattle. And we had samples at baseline and post-vaccination. And importantly, we also had matched data on protection in those animals following TB or mycobacterial challenge. So we could relate the levels of protein reactivity to the level of protection. So I'm not going to show you all the data because it's quite a lot of it. But this is just a snapshot to show you what came out. So this is non-human primates. So if you look at the left-hand graph, you've got baseline on the x-axis and post-vaccination on the y-axis. If your response at post-vaccination is proportional to baseline, then obviously you'd see a tight clustering of the points around the line of best fit. What we're interested in is these colored points that really are outliers because they have a much stronger response post-vaccination than would be expected given their baseline response. And then similarly, same data but plotted slightly differently on a volcano plot where you've got post versus pre-vaccination on the x-axis and the p-value on the right. These are the ones that are of interest. Similarly in the humans, it was slightly more difficult to see those proteins that really stood out here. We think it's because the humans had fairly strong baseline responses, which is probably due to pre-existing exposure to non-tuberculous mycobacteria, which then masks the response you get to BCG. This is the same data but plotted slightly differently, and it just allows you to see on the heat maps. So on the left of the heat maps is baseline, on the right is post-vaccination, and green, bright green is the lowest level of response going up to bright red being the highest level. And it just allows you to see that in the non-human primates at the top, you've got relatively good distinction between pre and post-vaccination, but in the humans it's slightly muddier, it's difficult to see that difference. Because they already have this pre-existing response. And the Venn diagram on the right I think is quite interesting because it shows out of the top 10 ranked proteins for each species, there was overlap in only one. So it suggests that actually different species have different target antigen repertoires for their antibodies. This is comparing the intradermal versus the intravenous BCG vaccinated non-human primates. And you can see the distribution here is quite different. Which is interesting because it means that the response you have to a particular protein following an ID BCG vaccination is not proportional to response to the same protein following IV. And so we're really interested in these ones that had a much higher response in the IV vaccinated animals than the ID because the IV vaccinated animals are much more protected. And you'll notice this protein on the left, which comes out as ranked very highly in terms of differential response, but actually this response to this was much higher in the intradermally vaccinated animals, and the intravenously vaccinated animals who were highly protected had a very low response to that protein. So that suggests that actually you could potentially have a detrimental antibody response here. So again, showing this as a kind of heat map representation. I quite like this because you've got the different proteins are rows on this heat map. The columns are individual animals. And if you look at those two columns on the right that have very high responses, lots of red, high responses to those proteins, they were actually the most protected animals. So if you look at their pathology, they had no pathology at all. They were sterilely protected. And that protein that I pointed out that maybe had a detrimental response is this one here. So you can see that those two sterilely protected animals had a very low response to that protein. So once we collated all this data, we went forward to select proteins that we were interested in, and we had a number of criteria for that. We were interested, obviously, in ones that had a large amount of had a good differential reactivity between pre and post-vaccination, because we didn't want ones that were just a pre-existing non-specific response or cross-reactive response. We wanted them to be responses induced by the vaccine. Obviously, we wanted that response to be associated with protection following in vivo challenge. We were interested in ones that were recognized across species, so this feeds into that One Health approach and the possibility of making a vaccine that could protect humans from MTB, but also cattle from M. bovis and humans from both MTB and M. bovis. And for that reason, we also wanted proteins that had a high degree of homology between MTB and M. bovis. For obvious reasons, we wanted ones that had lack of homology with host proteins, and then we're also limited by ones that we're able to successfully synthesize, because not all proteins lend themselves well to our expression systems. So just a slight detour here to show you the method that we use to screen. So this mycobacterial growth inhibition assay is something I developed during my PhD. It's a functional ex vivo assay that aims to be a correlate or surrogate of in vivo protection. And it means that you can do high throughput screens without having to challenge animals with MTB every time. And so this data actually came from the NIH, where they were validating this assay. So a higher level on the y-axis indicates superior protection. And on the left-hand side is data coming from a standard MTB in vivo challenge model in mice with different vaccine candidates. And on the right is using the MGIA assay, the in vitro assay, as a screen. You can see it's not quite as sensitive, but it does give you some idea of relative ranking, and it means that you can high throughput screen and find candidates that look promising and then take those forward for actual in vivo challenge experiments. So once we'd selected the proteins, we went on to start evaluating them using this model. We did this initially as just protein and adjuvant, and BCG was used as a positive control. It's very difficult to improve upon that in a mouse model. It confers very strong protection. But what we're really looking for is significance relative to a naive unvaccinated group. After correction for multiple comparisons, we did see that with one of these proteins, and then in the second experiment with 2 further candidates. And so we're interested in following up on those and seeing if we can improve on that protection using different strategies. So in conclusion, progress has been made in TB vaccine development in the past 20 years. Some new approaches are novel, some are things that we're revisiting. So these include things like improving or replacing BCG, some heterologous boost vaccines. But what we really need to understand is, like Lisa was talking about earlier, what are the correlates of protection? We've got a similar problem in TB as malaria, in that we don't know what those are, and Iman's going to tell us more about that in a moment. And then finally, using this proteome array approach, we've been able to identify specific proteins that are recognized by BCG-induced IgG, and it's been interesting to see that the route of administration of BCG seems to alter that target antigen repertoire. And this was a pilot study, but it does provide proof of concept for this approach to antigen discovery. And although I recognize that it's unlikely that it will be a purely antibody-inducing vaccine that will be effective against TB, I think if you can harness and combine induction of humoral immunity with a good cellular response, that you might be able to see better efficacy than we've seen to date using only the cellular response. So just to thank everybody involved in the protein array work, and happy to take questions.
Thank you very much, Rachel. So if you'd like to ask a question, could I ask just you raise your arm and we'll try and get a microphone to you.
Welcome in Egypt, and thank you for the nice presentation. I would like to ask about the aerosol route for vaccination. I can understand if we have a treatment through the aerosol route, which will be more logic. But for vaccination, how we can make sure that the amount of the drug that delivered systematically will be enough to create the antigen required for creating the immunity? This is first question. The second question, for the neonates, what is the best way to deliver the aerosol for the neonate? You know, the aerosol can be delivered through DPI, MDI, nebulizers. What is the best way to deliver aerosol vaccine to neonate? Thank you.
Yeah, thank you. They're good questions. I would probably leave it to the expert, Iman, because she's going to, the next talk, she's going to talk a lot more about aerosol, and she's been working directly on that. But in terms of standardizing the dose, I think the methods have improved a lot. So these nebulizers, although you do lose some of the dose through the process, it's possible to quantify the amount lost and then give a standardized amount, particularly in humans. It's harder in the preclinical models because it's hard to standardize what the animals are doing in terms of the way they're breathing, and especially if it's given under anesthetic. But in humans, you can control a bit more how they're breathing, how they're taking it in, and quantify exactly what's been taken in. So it's more standardized, but I will allow Iman to answer. Probably she can answer more thoroughly in the next question, in the next session. The second question, sorry, I've forgotten what the second.
I mean, for the delivery through the aerosol, how we can make sure that there is enough antibodies formed in the systemic system through the inhalation route?
Yeah, good question. So again, Iman will talk about this, but we've been quantifying immunogenicity both in the lung compartment and systemically in the periphery. So in the volunteers in the study, they get a bronchoalveolar lavage, so they get a sample taken from the lung, and then we look at immune responses in that as well as in the periphery. Thank you.
Thank you so much for this presentation. It was so informative, and it was a perfect attitude from you. I have a question. Actually, we've heard about BCG vaccine a lot in the past or in the last 2 years in the COVID pandemic, and we've heard about that this vaccine is connected to COVID-19, or we can use this vaccine to vaccinate ourselves against this virus. To what extent this is right?
Yeah, good question. So there was a lot of speculation about this through the pandemic because BCG confers some non-specific protection against unrelated infections. The idea was that it could perhaps be used as a stopgap until COVID-specific vaccines have been developed. And there were several studies, several epidemiological, ecological, correlative studies, which were a bit dubious. But actually, it's good timing because a big double-blinded, case-controlled, placebo-controlled study came out last week in the New England Journal. So this is the BRACE study coming from Australia, where they looked at healthcare workers. So they were randomized to receive BCG or not, and actually they found no efficacy of BCG against COVID infection or hospitalization. So I think that's probably a pretty good, strong answer that I think will probably put to bed the speculation on that.
Thank you so much.
Thank you.
Thank you very much, Rachel. And my colleague Ahmed is going to introduce the next speaker. Thank you.
Our next speaker is Dr. Iman Sati. Iman works as a senior scientist, and she's leading the clinical group in tuberculosis. And she's trying to develop different vaccination strategies for TB, like aerosol delivery of vaccines. And they develop control challenge model for TB and COVID as well, SARS-CoV-2. So thank you, Iman, for joining us.
Thank you. Assalamu alaikum. Hi, everyone. First of all, I would like to thank the organizers and Dr. Salman for giving me the opportunity to present today. And it's my pleasure to be back to Cairo, a city in which I spent the first few years of my life in. So today I will be talking to you about TB again, following on from Rachel's talk. And I will be talking about the insights we can gain into mycobacterial immunity when we analyze correlate samples collected from efficacy trials or from controlled human challenge studies. Next. Next, please. Okay. So TB is a complex pathogen, and we are dealing with a pathogen that evades the immune system. And clinical trials, especially efficacy trials, are complex, they are expensive, and they take a long time to complete, and they are affected by the disease incidence and also the logistics of sites, infrastructure in endemic areas. So when it comes to TB vaccines, we are not even sure whether a whole organism vaccine will be more effective like BCG, or a subunit vaccine in which you can combine a few antigens out of the 4,000 known antigens. So, and also problems facing vaccine development are mainly related to the lack of validated correlates of protection and the uncertainty about the predictive value of animal models. So 10 years ago, we completed the first efficacy trial using a subunit TB vaccine, and that was the trial that Rachel mentioned in her talk, the MVA-85A efficacy trial. So this is a viral vector vaccine which was developed in Oxford, and it was tested in an efficacy trial in South African infants. And these were it was a randomized trial. Infants were were either vaccinated with MV85A or received placebo as control group. So the study had 90% power to detect 60% improvement over BCG alone. So the outcome of the study was mentioned in Rachel's talk. So in this study, two outcomes were measured. One of them was immunogenicity, and although MV85A induced significant immune responses following vaccination, and this was measured as ex vivo interferon gamma responses to the vaccine antigen, which is antigen 85A. Although these responses were enhanced, the vaccine did not improve protection over PCG, of that induced by PCG. So, although the vaccine did not induce protection, samples collected from this trial were very valuable material, and they were used in studying correlates of protection. So in this study, infants were followed up for 3 years, and samples were collected before infants were vaccinated and after they were vaccinated. So for studying correlates of protection, we used samples that were collected before vaccination or at baseline. We studied immune responses at baseline and then linked it to the outcome of vaccination. So these infants were followed up for 3 years. During this time, some of them developed TB disease, some of them were infected with MTB and became latently infected without progressing to TB disease, and some of them remained healthy, and we refer to them as controls. So first of all, we studied correlates of risk of TB disease, and we collected those. We picked up samples from TB cases, and we had 71 cases. And we also used controls at a ratio of 1 case to 3 controls, and these controls were matched. And we used those samples to look at a range of immune responses, looking at cytokines, looking at chemokines, antibodies, and we also looked at the cell's capacity to control mycobacterial growth. So what we found, major findings in this study, is we found that activated CD4 T cells, so these are CD4 cells that are expressing HLA-DR, and these were not antigen-specific T cells. So these cells, the increased frequencies of these cells were observed in infants who got TB disease during the follow-up period. And then we also found out that BCG, specific interferon gamma, measured by ELISpot, was linked to increased protection or less risk of having TB disease. So these were the two major findings, and the third one was related to antibodies, and that was a surprising finding, because as Rachel mentioned, the role of antibodies in TB has been ignored for a really long time. So what we found in this study is infants who had higher levels of mycobacteria-specific antibodies at baseline to start with, they were protected from getting TB disease. So that was one of the major papers highlighting possible role of antibodies in protection. So, so far, I mean, what I presented so far is looking at mycobacteria-specific responses. So these are responses measured in response to mycobacterial antigens. But since our infants lived in a TB endemic area, exposed to a range of other infections, including viruses, we know that they were all HIV negative, so this is excluded. But we cannot exclude the possibility of exposure to other viruses. So we looked at immune responses to CMV and EBV viruses. So what we have done here was we used CMV peptides to measure interferon gamma responses to CMV, and then we dissected our infants, the cases and controls, by their CMV reactivity. When I say CMV reactivity, this is whether they were CMV positive based on their cytokine responses or whether they were CMV negative. And as you can see here, the proportion of cases, so cases are infants who got TB disease, was higher in the CMV positive pool. So not only this, but also these infants developed TB disease quicker than the CMV negative infants. So we also did a gene expression analysis, and we did look at transcriptomic correlates of risk of TB disease. And what we found, we identified 16 gene signatures that were differentially expressed between cases and controls. And these genes were defined when we looked at all cases versus controls. When I say cases, are TB disease versus controls, but also because we saw more susceptibility to TB in the CMV infected infants, we reanalyzed the data while dissecting the infants by their CMV status. So we have CMV positive infants divided into cases and TB cases and controls, and the same is for CMV negative infants. Looking at this, in the CMV positive infants, we saw downregulation of IL-23 and some NK cell markers, so killer-like receptors. And IL-23 is a cytokine that is involved with maturation of monocytes and macrophages and is known to play a role in control of TB. And in CMV negative infants, we saw an increase in T cell activation markers, including LAC and VCAM1, and together with other markers. And we saw the same in the when CMV negative and CMV positive infants were combined in this analysis. So, so what we have looked at so far is correlates of risk of TB disease. But then we also wanted to look at correlates of risk of MTB infection. So these, like infants who were infected with MTB but did not progress to active TB disease. So why do we look at MTB infection? What is the significance of that? What is the importance of that? So this is mainly because recently there has been a shift towards prevention of infection vaccines rather than prevention of disease vaccines. So the reason for this is because, like, if the trial is designed to detect prevention, is to detect MTB infection, then it's easier to get more endpoints because incidence of infection is higher than incidence of disease. So this will make the trial shorter and easier to conduct. So we did look at correlates of risk of MTB infection, and infection was measured by quantiferon test. So quantiferon is a test that measures interferon gamma in response to some TB antigen, mainly ESA6CFP10, and it's a measure of latent infection. So about 35%, so measurement was done at 3 time points, day -7, and this is the baseline, day 336 of the study, and then at the end of the study. So apparently all our volunteers were negative at day -7, and then because volunteers were selected based on them being negative to have negative responses to the quantiferon antigens. And then some of them, 35% of them, converted to positive quantiferon at day 336. And towards the end of the study, the majority of those which converted at day 336 reverted to negative. The rest of the volunteers in this study converted at end of the study. So first of all, what we looked at was to look at the previously identified markers or correlates of protection in TB disease. So one of our primary objectives of this study was to see whether the correlates of risk of TB disease are also correlates of risk of MTB infection. And then to do this, we looked at the 3 markers that we identified, which were activated CD4 T cells, interferon gamma, for BCG-specific interferon gamma, and antibodies. So what we found was the first 2 markers were not associated with increased risk of MTB infection. When it comes to antibodies, we saw a strong association between increased production of antibodies at baseline and susceptibility to TB, but the association was in the opposite direction in this case. So if you remember in the disease study, more antibodies at baseline meant less TB, but what we have seen here is more antibodies at baseline is more susceptibility to MTB infection. So we also looked at an array of cytokines, chemokines in plasma samples. So these are non-antigen specific. And we could see upregulation of inflammatory cytokines, chemokines, complement component factors, including, all this included interferon gamma, TNF, GCSF, and a number of inflammatory cytokines. So all these were elevated in infants that went on to become infected with MTB. So similar to what we have done in the previous study, we tried to look at the association between CMV infection and MTB infection. So in this case, we measured CMV infection by 3 methods. One of them was to look at CMV-specific antibodies, and we did look at IgG and IgM. So when we looked at IgG, we measured this at 2 time points, at day -7 and day 28, and to exclude any possible maternal antibodies, like crossing over and giving false positive results. What we have done is we only included those who have positive IgG at day -7, only if they maintain their positive 90% of their positive responses at day 28. And so what we have done is we considered any infant who is positive in any of the 3 methods used for measuring IgG CMV responses as positive. When we looked at the prevalence of MTB in the CMV positive pool and the CMV negative pool, there was a trend towards increased numbers of cases or MTB infection in the CMV positive pool, but this did not reach significance. So again, we have done gene expression analysis on those samples. So each row here represent a gene set, and what we can see in the columns is we analyze the data is when we combine CMV when we did not pay attention to the CMV status, and then we dissected the samples by CMV status. And we also looked at CMV genes that were differentially expressed in CMV positive and CMV negative, regardless of their MTB infection status. The reason for doing this was to try to look at whether there is any overlap between the genes that are differentially expressed when we looked at the MTB infection and the CMV infection, and tried to link these and to see whether susceptibility to to TB is linked to susceptibility to CMV. So when we look at the MTB positive negative, we saw upregulation of B cell and immunoglobulin genes and complement activation factors, upregulation of these genes in infants who became infected with MTB. And this is in line with the increased antibody secretion in this group. And then we also saw downregulation of neutrophil and activation and NK cell markers genes. So, and looking at the gene expression between if we just look at CMV positive versus the MTB positive, MTB negative in the CMV positive pool, and if we compare these to the same groups in the CMV negative pool, we see like very striking different gene expressions among these two groups. And when we looked at the overlap between the gene expression in the CMV positive versus CMV negative and MTB positive and MTB negative, the genes that were differentially expressed were mainly immunoglobulin and B cell genes. So it links to these B cell responses that we detected at baseline. So this is one way of looking at correlates of protection to conduct an efficacy trial, collect samples, and then look at baseline immune responses, and then dissect the results by infection or disease versus controls. But one problem with this kind of studies is it's impossible to control for natural exposure. So these infants will have different degrees of natural exposure. Although we do our best in like excluding infants or like volunteers with who are household contacts. So if one member in the family got TB, then that infant is excluded from the study because clearly this is increased risk, but we cannot 100% control for exposure. A better way here will be the controlled human infection model, and this is timed infection. So we deliberately infect people, and this is similar to what is done in other like diseases. But in TB, we are in a different position because it's unethical to infect humans with virulent MTB. So an approach we found around this that we developed an in vivo challenge model in which we use BCG as a surrogate for MTB based on the large homology between the two Mycobacterium species. So in this case, BCG is not used as a vaccine in terms of the readout, so it's used as a challenge agent. So our readout will be quantification of BCG recovery. So when we developed this model, we started by intradermal BCG. So BCG was given intradermally, and then skin biopsies were collected from the site of BCG infection, I would say, not vaccination. And then BCG is quantified in those skin biopsies. So BCG was quantifiable up to 4 weeks after vaccination. And then we used 2 weeks as our chosen BCG detection point because we saw a range of responses, reasonable range of responses. And then what we did on the figure on the right-hand side is we tested it in a different sets of studies. So we compared BCG naive individuals to BCG vaccinated individuals and those who received MV85A. So what we could see was that BCG vaccinated individuals could better control replication of BCG at the site of injection. And as you can see here, group D is our BCG MVA vaccinated, and these were not BCG replication in this particular study group was not different to BCG replication in the BCG naive group, which is in line with the outcome of our efficacy trial when this vaccine, the MV85A, did not show protection in an efficacy trial. So then we went further and we made more improvement to the model, and then we started delivering BCG via the aerosol route, and that's by inhalation. And we have 2 studies. One of them is completed, TB41 is completed and is submitted. TB43 is still, follow-up is still ongoing. So in those studies, the first study aimed at comparing immune responses in volunteers who received intradermal BCG and those who received aerosolized BCG. The second one, we had a group that got aerosolized BCG, and we had a control group that received aerosolized saline. So in both studies, our primary objective was to assess safety, and then also the second object to assess safety, and then the secondary objectives were to look at BCG recovery in the lungs and also to study cellular and humoral immune responses. So the idea here is to study immune responses in the lungs and also systemic immune responses, and to link this to how much BCG we quantify on those lungs and on samples collected from the lungs, and then to try to look at possible correlates of protection. So on today's talk, I will focus on the first part of these studies, which is the cellular and humoral immune responses. Work on quantification of BCG recovered from the lungs is still ongoing. So samples collected were bronchoalveolar lavage samples and peripheral blood samples. So looking at bile samples or lung mucosa samples, what we are seeing here is cytokine responses from CD4 and CD8 T cells, and we can see that in fact both routes of infection induce measurable responses, measurable cytokine responses, and as expected, the aerosol inhaled route of infection induced significantly higher cytokine responses in the lungs. So then we also looked at systemic immune responses, and surprisingly, the aerosol route of infection was as potent as the intradermal or systemic route of infection in inducing systemic immune responses. And the same was in the other study when we compared the controls and the when we compared the controls and the aerosolized BCG groups. So what we looked at also, not only cellular immune responses, but we also quantified antibodies. We looked at IgG and IgA, both in serum samples and in bile samples. So IgG, systemic IgG, was mainly induced by the systemic vaccination or intradermal vaccination. But when we looked at IgA responses, these were equally induced in serum samples. These were equally induced by the aerosol inhaled and the systemic routes of infection. There were no significant differences in bile antibody responses between the 2 routes of infection. So we also looked at different cell population in bile samples, which are mucosal samples, and compared these to whole blood. We saw an increase in granule size, and these are mainly eosinophils and neutrophils in bile samples, and we saw a decrease in antigen-presenting cells and activated antigen-presenting cells in the lungs. So when I say antigen-presenting cells, these are collective monocytes, macrophages, and monocytes and macrophages and dendritic cells. So we looked at NK cells, and we saw an increase of NK cells at day 7 following BCG infection. Initially, we expected NK cells to peak a little bit earlier, but it seems like in the lungs, it peaked at day 7. Not too much is going on in NK cells in peripheral blood. So we also looked at T cells, and these are total T cells, CD3 positive T cells, and we saw an increase in this population in the lungs at day 7 following infection, and In blood, we saw increase in this population all the way up to day 56. So in blood, we have the opportunity to look at the kinetic of the immune response because we collect blood samples repeatedly during the study. When it comes to mucosal immune responses, we are just able to collect a single bronchoalveolar lavage sample. So dissecting the T cells further into CD4 positive and CD8 positive subsets, we see increase in these populations, not only in the CD4 positive, CD4 negative, and also in the double negative cells, and in the lungs we saw a decent population of CD4, CD8 double positive cells. So these were just cell populations, like phenotype being cells, and this wasn't antigen specific, but we also looked at antigen specificity, and we assessed interferon gamma to BCG. So we did an in vitro stimulation where we stimulated bronchoalveolar lavage cells with BCG. And we also looked at non-stimulated cells because the theory here is there is BCG in the lungs, so this is considered probably as in vivo stimulation. And as you can see, we see induction of CD4 and CD8 cytokines in the lungs, and we also saw some sort of induction, some level of cytokines in non-stimulated cells, but we can still also see some response in the controls. In this case, we cannot exclude exposure to other source of probably environmental mycobacteria. So in summary, immune correlate samples from efficacy trials allows like studying further dissecting immune responses and to allow better understanding of protective mechanisms. And what we have shown in our correlated studies that mechanisms that control TB infection are different to those that control development of TB disease. So we need also a human challenge model that is optimized to facilitate detection or studying of correlates of protection. And aerosol route of delivery would allow studying immune responses, local immune responses, or like mucosal responses, in addition to systemic responses, and try to link this to outcome of infection. So by this, I conclude my talk, and I would like to thank everyone in Helen McShane's group, and I would like thank the funders, and most importantly, all the study participants, and thanks for your attention. I'm happy to take any questions. Yeah, there was a question about, yes, your question about BC, how can we make sure that we deliver the right quantity of BCG, right? So we do quantification for BCG. So what we do, for the intradermal or systemic BCG, we use the standard dose of BCG. But then when we started to optimize this method for the aerosolized route, we used nebulization. So what we have done is we put BCG through the nebulizer, and we quantify what we get at the other end. And when we did that, what we found was we lost 1 log of BCG through the nebulization process. So we ended up giving, the dose was higher through the aerosol route. It was 1 × 10⁷ compared to 1 × 10⁶ when we give it intradermally. But in reality, it was the equivalent dose to what was given intradermally, because we know that we lose 1 log through the nebulization process.
So you measure what comes out of the nebulizer?
Yes.
But during the reality, half of what come out of the nebulizers is only inhaled, not only half, 1/3, because I inhale for 1/3 of a minute and exhale in 2/3 of a minute. So out of what comes of nebulizers, only 1/3 of this reach the lung, not reaches the lung, reaches the patient. And out of this 1/3, there's only, most of it hit the throat, and a little bit reach the lung because the diameter of the droplets. So how do you identify exactly the dose that reaches the lung?
We cannot identify exactly that all of it reached the lungs.
Yes.
Because, I mean, all these are factors,
Yeah.
I mean, all these are steps during which BCG can be lost. What we can control is how much comes out of the nebulizer. But the way one human breathes might be different to another one, but we clearly cannot control for 100% we cannot measure how much BCG is exactly in the lung. All what we can do is when we take that fluid is to quantify the BCG that we get in that fluid. But how much is lost, this is beyond our control.
There is some in vitro techniques to do that. Maybe you can use like imbectors or something like that to measure yeah. But my other question, is this nebulizing system suitable for neonates or infants?
It has been, I mean, so far it hasn't been used in infants, but we use it in non-human primates. We use it in, I mean, I'm not sure whether this is the right comparison between infants and non-human primates. I'm not sure how feasible that is in very young infants, but it's a deployable method, it's needle-free, it's pain-free as well. Yeah, but when it comes to the quantification of BCG, considering all the possible losses that happen on the way, and that we are still able to detect those immune responses.
Yeah, that's amazing. I understand that. But
so, yeah. Regardless of how much is being lost, because there is a limit to how much we can increase it.
Yeah.
We can increase it by 10-folds, but remember our primary readout, our primary, like, we want to make sure that it's safe. It's not only about the induction of the maximum, like, possible immune responses, but it's also about safety.
Yeah, what I am saying that we can reach a higher delivery with the aerosol systematically.
There is another way. I mean, like, a group in South Africa deliver BCG by instillation. So if it's delivered by instillation, then it's given directly into the lungs. But how practical is that in reality? I mean, if eventually people decide that the aerosol route is the best for delivering this vaccine, will it be feasible to get people and do instillation and put BCG directly to the lung? Which one is more feasible? Or will it be easier to give people an inhaler, even if it's infants, and just do it through inhalation? So it's a balance here.
I am with the inhalation. I am not with but I mean there's still a window for more improvement. Because this is a very promising
I agree 100%. There is a way to improve this. There is a lot that we need to do in, like, a bit more standardizing the method. Because, like, so far all what we could do is two things. First of all, to know for sure how much is coming out of the nebulizer. The second thing that we could do is to look at how much we recover in the ball. So these are the two things that we can control now, but there is a window here for improvement. There is a window here for finding a way to better, to better standardize what is being delivered. Definitely, I agree with that. Yeah.
Thank you.
Thank you. Any more ideas will be really helpful in that as well.
Fantastic discussion there. Any other questions, please, for Iman? No? Full service.
Thanks for your presentation. I just want to ask about the relation between the CMV virus and the development of TB. Is just specific for CMV virus that is compromised the immune system, or other viruses can also affect the TB infection? This is my question.
Yeah, I mean, in fact, I mean, in our, we didn't only look at CMV, we looked at CMV and EBV, and we saw that association. And I agree there might still be, like, association with other viruses. I mean, other studies published this. In our study, we only looked at these, but it's not necessarily these are the only ones that are associated with susceptibility. Yeah.
Okay. BCG vaccine is only specific for some populations and the other is not, or it is available for all populations?
Population.
It's the effect. It's the effect of the BCG vaccine.
Yeah.
It's specific only, population.
So BCG is the WHO licensed vaccine. It's the same vaccine that is delivered or available for all populations. But when it comes to its efficacy is different in different parts of the world. It gives different degrees of protection, and we know that in TB endemic areas, protection from BCG is minimum. So the range of BCG efficacy or protection ranges from 80% protection to nothing, to zero protection. And this zero is mainly in TB endemic areas. So we don't know whether this is linked to the genetic makeup of these populations, whether it's linked to nutritional factors, whether it's linked to exposure to other mycobacteria, but we know that in certain populations, BCG protects better than others, but it's the same vaccine that is available for everyone.
Its side effect is only variable between populations or not?
When it comes to side effects, it's comparable. I'm not aware of any reported differences of BCG side effects based on which population is which.
Okay, thank you.
Okay, thank you.
No worries.
Thank you, Dr. Ben. Thank you for the
Thank you.
presentation. Then we have to invite our next speaker, Dr. Berna Boutar. She is a doctor in oncology, graduated from Aix-Marseille University, France, 2020. She is interested in understanding the interaction between cancer cells and immune cells. Her PhD project aimed to unravel the mechanisms of evasion of natural killer cells, cell-mediated immune defense in acute myeloid leukemia. She is also worked as a researcher associate at the University of Cambridge, where she studied the immune response in ovarian cancer. Also, she is currently working as a research associate at the University of Oxford. Her aim of research is to find ways to potentiate the natural killer cells for the cell therapy against hematological malignancies. Okay, welcome, Dr. Berna.
Thank you.
Here is stage with you.
Thanks for the introduction. Thank you all for being here today, and thanks for the organizers and specifically Dr. Ahmed, who did the link between us in Oxford and this conference here. I'm gonna change a little bit the subject, as I don't work on infections and I don't work on vaccine. I work on cancer, but still in the immune system. Okay. So cancer is a disease that doesn't need introduction nowadays. However, I just wanted to say this historic piece of information just because we're in Egypt today. That the first evidence of cancer was found 3,000 before Christ in fossilized bone tumors and in ancient Egyptian medical books. And they characterized cancer as ulcers that cannot be treated, and that they had to be removed. So now this cancer does have treatment, and the removal or the surgery is one way of treating it. The other ways are the chemotherapy and radiotherapy, which are very traditional way to treat cancer. These treatments are efficient, but they are also invasive, and they are toxic for highly proliferative cells that are not cancer cells, which is due to their unspecificity. And even though there has been some efforts into making these treatments less toxic with minimally invasive, better dosing of chemotherapy and using of precision radiation, there's still some toxicity and recurrence of the tumor because of the drug resistance, because of the tumor heterogeneity and the presence of quiescent cells that could always develop tumor again, and because of the metastasis of the tumor and the difficulty to trace all the sites. And because of that, the clinicians and the scientists started to look for alternative therapies, and two main approaches have revolutionized the treatment of cancer. The targeted therapy, which is targeting signaling pathways important for cancer survival or proliferation, and the immunotherapy. So I'm gonna focus more on the immunotherapy part. The immunotherapy is not new. It has started in the 19th century with some correlation studies between infections and the appearance of cancer, but it became more convincing in the last 20 years, and it has made the cover of important journals in the 2012-2013. Very briefly, we know that we have different immune cells in the organism that could be used to fight against cancer. These immune cells can either be innate immune cells, so first line of defense, or adaptive immune cells that take more time to develop, but they are more specific of the pathogen. And for a long time, the focus of the cancer immunotherapy has been on the adaptive immune cells and specifically on T cells. And this is because of their cytotoxic capacity, their capacity to eliminate tumor cells, and because of their abundance in the organism. So what are the strategies that have been used on T cells? First, just to remind you very quickly how T cells work. T cells have a specific T cell receptor, or TCR. It can recognize a peptide linked to MHC molecule, either presented on antigen-presenting cells or on tumor cells. And it also expresses co-signaling molecules that upon binding with ligands, it's gonna give a signal for T cells to be even more activated. And once T cells bind to, I lost the point, I'm sorry, once T cells bind to their antigen and in the presence of the co-signaling molecules, after their activation, they will start expressing inhibitory receptors. And this is to resolve the immune response and to avoid having any aberrant and overreacting immune response. And so these inhibitory molecules can either be already produced and present inside the cytoplasm or can be produced de novo upon activation of T cells. And cancer cells express a lot of these inhibitory ligands that can inhibit T cell activation. And so one approach to activate T cell response more in cancer was to use antibodies against these inhibitory ligands. The most famous one are PD-1 and CTLA-4, which led to Nobel Prize of Medicine in 2018 for the two main researchers who worked on these molecules. Another way to activate T cells is the use of adoptive cell therapy and mainly CAR T cells, in which T cells are genetically edited to express chimeric antigen receptor that can help recognize tumor cells and eliminating them. One approach is also the use of the vaccine, and I'm talking about the approach today mainly because it's a vaccine workshop. So the vaccine can be either prophylactic to prevent the appearance of cancer, or it can be therapeutic. It means that patients have already cancer and they are vaccinated to boost the immune system against a specific tumor antigen. So these approaches have been successful and have led to FDA approval of many of them as monotherapy or combination therapy in several cancer indications, but they also have their limitations linked to the adverse effects of using T cells, such as cytokine release syndrome or neurological toxicities. Also, cancer cells can avoid the recognition by T cells due to the loss of antigen expression or the loss of MHC class I expression. So this has led to the need to find other alternative or complementary approaches to T cell therapy. Another reason for using alternative and complementary approaches is the fact that T cell responses are not autonomous. They need the priming and they need the cytokines produced by the innate immune system. So it became more evident that there is also a need to harness the innate immune system for cancer therapy. And as I said, one reason is to prime and develop even more the adaptive immune response. And as example is the use of agonists of innate immune cell receptors, such as TLR9 agonist or STING agonist, which gonna activate innate immune cells. They're going to secrete cytokines and activate T cell infiltration. These could be also added in addition to T cell treatments. Another example are approaches to increase the maturation of dendritic cells. Another way to, or another reason to use the innate immune system is for itself being able to eliminate tumor cells. And innate immune cells such as macrophages and NK cells have receptors for this constant FC part of antibodies. So they can be directed by the use of monoclonal antibodies to the site of tumor, and they can eliminate these tumor cells either by cytotoxicity or by phagocytosis. NK cells are an example of innate immune cells which don't need, can be activated by the antibodies, but can also recognize tumor cells without any need for antibodies, and they have a cytotoxic capacity and are able to eliminate tumor cells. So I'm going to focus today more on natural killer cells. So natural killer cell functions mirror totally NK cell functions, and to explain their functions, I'm gonna again go through T cell functions. As I said, T cells have receptor. It can recognize MHC class 1 molecule with its peptide, and co-stimulatory and co-inhibitory molecules that can recognize also their ligands. After T cell activation, they will secrete cytokines and chemokines such as interferon gamma and TNF alpha. They will also release cytotoxic granules containing perforin and granzyme, which will lead to cancer cell death. NK cells, on the opposite part, they don't have any specific receptor for antigen. The receptors are not produced by somatic recombination. They just have germline encoded receptors, and a lot of them. So there is a big repertoire of inhibitory receptors and activating receptors that they express that I just simplified in this figure with one inhibitory receptor and one activating receptor. The inhibitory receptors are mainly specific for MHC class 1 molecules, and the activating receptors are specific for ligands that can be expressed on stress cells, such as virus-infected cells or cancer cells. And the way NK cells can be activated is via a balance of these activating and inhibitory signals. So in this case, for example, where NK cells is in contact with the healthy cell, then the balance leads to neutral. There's no activation of NK cells. However, if NK cells is going to recognize cancer cells increase the expression of stress ligands, so the balance of signals will lead to an increase in the activation of NK cells, and as you can see from this figure, the functions are exactly the same as T cells. They can kill cancer cells by perforin and granzyme B, and they can also secrete interferon gamma and TNF alpha. Now, when cancer cells lose MHC class 1 expression to avoid being recognized by T cells, they become even more visible to NK cells because the threshold of activation becomes lower, and even a low expression of stress ligands will activate and trigger NK cell functions. So NK cells could totally complement T cell functions when the recognition of tumor cells by T cells cannot be accomplished. NK cells are not, haven't been discovered very long time ago. They have been discovered in the 1970s, but as soon as they have been discovered and understood, they have started to become a focus of cancer immunotherapy, and today there are over 200 clinical trials using NK cells for cancer immunotherapy, and these are only examples of the clinical trials or of the approaches that could be used, like the activation of the NK cells of the patient ex vivo by antibodies, by cytokines, and re-injecting them into the patient, or injecting NK cells from a healthy donor into a patient, or even the use of CAR-engineered NK cells, which is the equivalent of CAR T cells. NK cell immunotherapy also faces many challenges, including their specificity. So they don't need a specific antigen, tumor antigen, but sometimes the recognition of stress ligands is also not very evident. So sometimes they need to be engineered to improve their recognition of tumor cells. They can also be inhibited by the ligands expressed by tumor cells. They can be inhibited as well by the very immunosuppressive tumor microenvironment. The homing of NK cells into solid tumors remains a challenge, as they're usually found very low infiltrating in solid tumors, and their persistence as well. So I'm going to focus more on the persistence. NK cells are short-lived cells. They live in humans about 2 to 3 weeks, and they are renewed after that. So for their use as immunotherapy, it has been considered more as a short-term therapy. So they are injected into patients, and they are not detected after 2 weeks. But some groups had this idea that if we can make NK cells express their own cytokines that are essential for their survival, we can make them survive for longer. And after this approach, it has changed the idea of NK cell immunotherapy from a short-term therapy, which is only for about 2 weeks, to a long-term therapy where NK cells could be detected up to 1 year after one single infusion. And knowing now that NK cell therapy is becoming more and more persistent, it means that NK cell therapy is facing a new challenge, which is the exhaustion. You probably have heard about the exhaustion from T-cell approaches, and you know that T-cell exhaustion in cancer is a major problem. However, NK cell exhaustion has long been neglected because of these cells used to not survive into the patients for a long time. So the exhaustion is a mechanism of dysfunction that happens into the cells when they are chronically stimulated, and it leads to a decrease in their functions. So in the case of NK cells, it's a decrease in their cytotoxicity, their cytokine secretion. They also change phenotype, and they increase the expression of inhibitory receptors, and they exhibit also stable epigenetic changes. It remains unclear if NK cells really get exhausted following chronic stimulation, as in cancer and infections. There has been studies that showed that NK cells, or that described NK cell dysfunction as an exhaustion. However, these studies are very heterogeneous, and they are not looking for any epigenetic changes in the cells. So we don't know what are the real characteristics of exhausted NK cells, and what are the regulators of this NK cell exhaustion. We would like to know if manipulating these regulators of NK cell exhaustion could prevent or could reverse this phenomenon, and if this could bring any benefit for cancer immunotherapy. So I am particularly interested in these questions, and this is the project that I am doing in Oxford. And to study the exhaustion of NK cells, I'm using a very simple system. So going back to the bases, NK cells are activated through interaction between ligand and receptor. So if I put cancer cells with NK cells together for a long time, I should detect if I have an exhaustion. I'm using cell lines for NK cells, the NK92 cell line. It's a cell line that has FDA approval to be used in clinical trials. And for cancer cells, I'm using a chronic myelogenous leukemia cell line, K562. So I tried to put NK cells with irradiated cancer cells together for 12 days, and I supplemented more irradiated cells at day 2 and at day 4. And then after 12 days of culture, I collected NK cells and I put them again in vitro with tumor cells, this time only for 4 hours, just to assess their functions, such as their degranulation and cytokine production, and to assess their killing capacity. And as you can see, when NK cells have been cultured with tumor cells for 12 days, which is shown here in gray, they decrease their degranulation, they decrease their interferon gamma production, and their TNF-alpha production when they are compared with NK cells that have been cultured alone during this time. They also have reduced killing capacity, which shows that all their functions are reduced. Now we know that cancer cells do more than just the activation of the cells. They can produce immunosuppressive cytokines, they can use the metabolites, they can use the nutrients, change the pH of the media. And even though we irradiated these cells, we wanted to make sure that what's happening is linked to the chronic activation. And for this, we tried a complementary approach, which is the replacement of tumor cells by antibodies that can activate the receptors. We decided to use two different stimulations. One is the antibody against the receptor NKp30, and the second one is the antibody against two receptors, NKp46 and NKG2D. We have shown that the stimulation with these antibodies can activate the degranulation capacity, the interferon gamma production, and the TNF-alpha production of NK cells in a short-term assay. Now we wanted to see what could the stimulation of cells with antibodies do for in a longer-term assay. So we put NK cells in culture with wells pre-coated with these antibodies. So as an example, NKp30 or the isotype control for a control condition. And we put them in culture for 2 weeks. And we collected samples of NK cells at different time points to see what is happening during this culture. And to see that, we cultured them with tumor cells and assessed their functions. So at day 1, which is an acute time point, acute stimulation of the cells, we could see either equal or an increase in the capacity of the cells to produce interferon gamma and TNF-alpha when they have been pre-stimulated with antibodies. However, during this chronic stimulation of the cells, we could detect a decrease in their degranulation, decrease in their interferon gamma production, and decrease in their TNF-alpha production. We could see the same effect using the second system, which is the stimulation of NKp46 and NKG2D. It also led to a decrease in the functions of the cells after chronic stimulation. We were also interested to see if there is any change in the phenotype of the cells, and for this we looked for the expression of surface receptors by flow. So the data that you are seeing here is just a fold change of the expression in the stimulated condition normalized to the isotype control condition. And we could detect at the acute time point majorly an increase in the expression of all receptors. But the more we enter the chronic phase, the more the activating receptors start to be underexpressed. However, the inhibitory receptors expression remained high. And this trend, with some exceptions, we could see it even with the stimulation with NKp46 and NKG2D, which shows that the activating receptor expressions start to decrease and the inhibitory receptor expressions start to increase after chronic stimulation. So to conclude on this part, we have shown that the chronic stimulation of NK cell decreases their functions and changes their phenotype. We still don't know if there are any stable epigenetic changes in the cells. What's important to know about the stable epigenetic changes is that they are responsible for making the cells poised in a dysfunctional state, even when the stimulation is removed. So we asked ourselves whether NK cell alterations could be reversed after we stop the stimulation. So for this, we used the stimulation with antibodies, but we changed the system. We stimulated the cells only for 1 week with the antibodies, and then we had 1 week of resting for the cells, though they were transferred in a well without any stimulating antibodies, to see if their functions could recover. And we could see at the beginning of the stimulation, at the acute time point, as I shown previously, an increase in the functions of the cells. When the cells enter the chronic stimulation phase, they start to decrease their degranulation and their TNF-alpha production. And interestingly, we showed that even when we stop the stimulation for 1 week, there isn't any recovery of NK cell capacity to degranulate or to secrete cytokines. Same thing goes for their killing capacity. So we saw a decrease in the killing capacity of NK cells when they are chronically stimulated with antibodies, and this difference could not be recovered after 1 week of resting. Now that we know that the alterations could not be recovered, we wanted to see with molecular techniques if there are any stable epigenetic changes. For this, we used the ATAC sequencing and RNA sequencing. So ATAC sequencing is a technique that allows to see if the chromatin is more open or closed, and RNA sequencing is for gene expression. I'm going to show only the data today on ATAC sequencing. Okay. First, by PCA comparison of the samples, Just by PCA comparison, we could see that the controls of non-stimulated cells cluster differently at a different place than the cells that have been stimulated after 1 day, which means there is a big difference in the chromatin accessibility between these two groups. Interestingly, the cells stimulated for 3 days and 7 days, which I hope you can follow with me. Yeah, they cluster also very much differently from the cells stimulated for 1 day, and they cluster almost together, which means they have almost the same chromatin accessibility and very different from the cells stimulated for 1 day. After 1 week of resting, we have also a different landscape of chromatin accessibility. And this, you can see it here also with this heat map. For the controls for the controls of non-stimulated cells, whether at the beginning or at the end of the culture, they look quite the same, but they look very differently from the cells that have been stimulated for 1 day, with more open chromatin for the cells stimulated for 1 day. After long-term stimulation, we start to see some regions that are closed in the genome. And interestingly, after 1 week of resting, some of these genes and these regions cannot recover and remain closed in comparison to naive cells. So these are still preliminary data. We still need to analyze what are the main genes and the regions that are open and closed during these states, which could help us understand the regulators of the exhaustion. But we have another approach as well to understand these regulators, and this is by the use of CRISPR screen. So maybe you are familiar with the genome-wide CRISPR screen to understand regulators of some mechanisms. In our case, we are doing an epigenome-wide CRISPR screen, which means we are focusing our screen on only 5,000 and around 500 epigenetic and epitranscriptomic regulators. So we have made a customized library of guide RNAs targeting these regulators. We have designed 4 guide RNAs for every gene and 1,000 of non-targeting control guide RNAs. And the idea is to make these NK cells express every NK cell express 1 guide RNA targeting 1 regulator only. So we will obtain a pool of single knockout NK cells. And if in some of these cells we have knocked out a regulator of NK cell exhaustion, then when these cells will undergo the chronic stimulation, we would be able to see that some of them would be protected from the exhaustion. We could sort these cells by flow and then submit them to sequencing to identify candidates for regulators of NK cell exhaustion that you would need later to validate and understand their mechanism. So to conclude, we have shown that the chronic stimulation of NK cells by co-culture with tumor cells or by stimulation with antibodies can lead to reduction in their functions and changes in their phenotype and in their chromatin accessibility. This work is still ongoing, so we still need to process some of the ATAC sequencing and RNA sequencing data. We aim to identify the regulators of NK cell exhaustion by CRISPR screen, to validate our findings in primary and human and mouse NK cells. And we're also interested in the therapeutic potential of manipulating these regulators. And this work, we focus mainly on stem cell-derived NK cells, specifically induced pluripotent stem cells-derived NK cells, because it's a model that is very close to the clinics. To finish, I would like to thank everyone that contributed to this work, also the collaborators in Oxford and in the US, and thank you for your attention.
Thank you so much for this kind of presentation. Now, open the discussion. Yes?
Okay, so thank you so much for your presentation. Actually, I have many questions because I'm so interested in this part. First, I'm talking about the stage of the disease. We know cancer has 4 different stages. So did you guys take it into account or take it into your considerations? That was for the first part.
You know, this work is very, very fundamental at this stage. It's just putting cells together in vitro, right? So it's not applied to any cancer stage yet. If it has to be validated in mouse models and models of cancer, they usually look more like stage 3 and stage 4 because otherwise they would not develop in the mouse models. But at this stage there isn't any cancer stage yet. It's very preclinical. Yeah.
Okay, perfect. So for the second part, I'm talking about the universal vaccine. Cancers have a lot of genomic instability. So how to design a therapeutic vaccine against cancer, or make a universal vaccine? Is that possible to make a universal vaccine against cancer?
I'm not sure I'm the person who's like the most has the biggest knowledge on the vaccine and cancer, as I don't work in vaccine and cancer. But what I know is it's ideally it would be a tumor-associated antigen that would never, would always be expressed on tumor cells and in different tumors, and in one case of cancer it would be expressed in all patients. But the reality is this could never happen, and so sometimes it's about an antigen that is expressed in a lot of patients but at low levels, or an antigen that is expressed at high levels in a few patients. Some also go to personalized vaccine therapy. So they take samples from the patient, they sequence them, they identify the tumor-associated antigen, and then they produce the vaccine, and then they inject them into the patient. So yeah, I mean, it's a bit of a broad answer, but...
So, it has nothing, it has a lot to do with personalized medicine, right? So the third part, I'm asking about the advantages that this approach offers. This approach is called, is based on stimulating the innate immune system to fight against cancer. So what are the advantages that this approach offers like T-cell therapy or anything else?
Yeah, very good question. So usually people go always to T-cell therapy, right? Because it's like what has shown to be the most efficient. So what could be the advantage of using NK cells? The advantage is that we are not talking about personalized treatment this time, because NK cells don't need recognition of any tumor-associated antigen, and they also can be injected from any source to the patient without any risk. So they could be injected from a cell line, or from stem cells, or from a healthy donor. So there is an easy way to find sources for NK cells that could be safe to inject into the patient, and they could be applied to a vast population without having to personalize the treatment.
All right. So the last question: how far we are to make it available? Available for the people. How far we are from that for immunotherapy in general?
It's not far at all, immunotherapy in general, because some are standard treatments now, like in melanoma and in lung cancer, the use of CAR T cells and the use of antibodies are standard. Also in B-cell lymphoma, the use of CAR T cells is very, very standard. For NK cell therapy, it's still into clinical trials, because also these cells, as I said, they haven't been discovered for a long time, so it needs some time to understand how they work and then to take it into the clinic. But there are many, many clinical trials and published studies on their efficacy, so it's possible that they get approval as well.
Okay, thank you so much.
Okay.
Thanks.
Thank you so much. Any further questions? It's okay. Thank you, Dr. Bernas.
Thank you.
Okay, thank you very much. And now I'd like to introduce Dr. Gulbus Turan to the stage. So Gulbus is currently doing her DPhil studies at the Jenner Institute in the field of vaccinology. She's actually medically trained, so she's already a doctor, so it's going to be a doctor. She works as a clinician during the COVID-19 pandemic before joining the Jenner Institute and doing her studies in clinical medicine at the University of Oxford. Her research focuses on developing innovative virus-like particles to combat the Plasmodium falciparum malaria we heard about earlier, and targeting the pre-erythrocytic stage of the disease, with the ultimate goal of improving health and well-being in those in the affected regions. And I'll leave it there, and we look forward to your talk.
Thank you very much. Thanks for staying to listen to my talk. I will be talking about my own PhD project in Jenner. This is already This is the Plasmodium falciparum life cycle. You already heard about that, and this is the deeper journey of Plasmodium sporozoites. So Plasmodium sporozoites are Plasmodium sporozoites deposited in the dermis of the mammalian host by the bite of an infected Anopheles mosquito, and those sporozoites are sequestered in the liver sinusoids through interactions between the circumsporozoid protein, which we already have, the vaccine, which is R21, and hepatic heparan sulfate proteoglycans. Then they cross the sinusoidal cellular barrier through liver sinusoidal endothelial cells or Kupffer cells. And this is the sporozoid's molecular model for sporozoid cell traversal. And as you can see here, there's some micronemes and rough trophy organelles apical end of sporozoids. And to produce some VLPs against our antigens, this is the schematic description of my VLP production process. We clone the targeted antigen plasmids into Escherichia coli bacteria, and then we use yeast expression systems to express proteins even more. And after that, we use high-pressure homogenization systems to help the VLPs to self-assemble themselves. After purification, we use CTEC affinity chromatography, SEC chromatography, Western blot conformation, electron microscope conformation. After those conformation steps, we have purified VLPs. And those VLPs are ready to formulate with adjuvant and excipients, and after that, administered to mice. This is some steps for size exclusion chromatography that we use for purification. And these are the Western blot results for some antigens we use for vaccine. And this is, you see on right, is electron microscope images for R21 vaccine. I compared R21 with Matrix-M and with a different adjuvant using this experimental design that I will talk about later, but I wanted to show it first. Because we know that adjuvants really important to produce even more immune response to that if we can combine them with good vaccines. These are the adjuvants used in human vaccines so far. The journey of adjuvants starts with aluminum salts, which basically since from 1930s, and now we have different type of adjuvants like virosomes, MF59s, adjuvant systems, oil and water emulsions, and immune stimulant complexes such as Advax and Matrix-M. As you can see here, they are the current licensed adjuvants for human use. And for R21, which is almost about to get approved by WHO, we use Matrix-M, which is a saponin-based immune stimulation, saponin-based adjuvant that produced by Novavax. These are the main adjuvant groups that we can say can be described within a spectrum of functions ranging from antigen delivery to immune stimulation. As you can see, nanoparticles, liposomes, water in oil emulsions, mineral salts, ISCOMs, oil in water emulsions, TLR agonists, and saponins. We can achieve even more immune response if we manage to combine these different adjuvant groups, like combining mineral salts, something else like TLR agonists with saponins and nanoparticles. For example, these are adjuvants from Vaccine Formulation Institute. They use liposome-based adjuvants and squalene emulsion-based adjuvants, which include cholesterol, Q-urea, saponaria extract, and TLR4 ligand to achieve more immune response. And this is the experimental design that I used to compare R21 low dose and standard dose, which is for low dose it was 0.1 microgram, and for standard dose it was 1 microgram. And for Matrix-M, I used 5 microgram Matrix-M to compare with all of them. For the other adjuvant, which is adjuvant X, I said adjuvant X because I don't want to explore because it's unpublished data yet. I use also 5 microgram adjuvant X to compare it with Matrix-M. During the experiment, I applied prime and boost vaccination, and after 3 weeks, we did challenge, and then we screened the mice to see if it caused any parasitemia or the vaccines prevented the mice from any parasitemia. These are the immunization and efficacy results after prime vaccination. As you can see here, group 1, 2 provided almost very close immune response, which is really good, and group 3 and 4 also almost the same. And these are BALB/C and CD1 results. We see very similar immune response between groups. As you can see here, these are the post-boot immune response results after ELISAs, and we find that there is no significant difference between adjuvants or doses, which means if we use lower dose of antigen, it can provide a similar immune response. So it may allow us to dose pair for even more antigen, even more vaccines. And also we find there is no significant difference between Matrix-M and adjuvant X, which means we can use adjuvant X if we need to use different adjuvants in terms of vaccine supply. And these are the efficacy results after challenge. It showed that in BALB/C mice, group 1 and group 2 showed very high efficacy, and group 3 and 4 also more than 50% efficacy. And in CD1 mice, they were also really similar, and we didn't find any significant difference between them, which means Matrix-M and adjuvant X were really similar effect. I'm trying to wrap up really fast because I know we already passed the time. So as conclusion, a lower dose of R21 showed similar levels of efficacy to standard dose, regardless of adjuvant type, suggesting prospective feasibility to deploy more vaccine doses with the same quantity of VLPs. And the preclinical vaccine efficacy of R21 can be replicated with alternative adjuvants, such as Matrix-M and adjuvant X, encouraging further evaluation to ensure steady vaccine supply. And adjuvant dose plays a key role in R21 efficacy according to results of a phase 1/2b study exploring the combination of R21 with a low and high dose of Matrix-M. Our results encourage the exploration of multiple adjuvant doses to further explore R21 efficacy improvements. And these are the acknowledgements. Thank you very much for staying and listening to me.
Thank you very much, Gulbus. Perfectly timed. We have
Yes,
time for
that's
some
good.
questions. Maybe I could start. So obviously you can't tell us what the adjuvant is, but I mean, how different in structure is the this adjuvant X to Matrix M? Matrix M is a nanoparticulate sort of cage-like.
Matrix M is immune stimulant complexes. So adjuvant X has additional TLR4 agonist as well. So adjuvant X effect use the nanoparticles, like saponin-based nanoparticles, and they also use TLR4 agonists. That's the main difference, and MPLA, yeah.
See, I only ask the easy questions, so. Would anyone like to ask any questions? It's been a long day, I know.
Thank you very much.
I could ask more questions, but I think we'll let you off. Thank you very much, Gulbus. Thank you very much. I'm going to ask Dr. Ahmed Salman to just wrap up this very interesting session. The floor is all yours.
Yeah, thank you so much all, and thank you for staying late till the end of the session. So just quickly wrap up and kind of give recommendations. Today, like this session was really focused in TB, which is still a major global issue. And I think TB is still the top killer. It kills over 2 million to 3 million every year. And it's more increasing with having the multi-drug resistant strain. So focusing in that and putting more effort in research, especially, although in Egypt there's of course a massive improvement in TB, but still it's a serious issue globally and in Egypt as well. And there's different way, of course, immune therapy and vaccines, still protection better than cure. So having no 100% efficient vaccines and varying efficacy based on races, based on an age, and is not protective after childhood, like the current vaccine, BCG, that's actually a concerning thing, and more research are needed. The malaria and adjuvants and better understanding, although in Egypt we don't really suffer malaria in maybe except very small areas, but with the global warming and also with having some resistance and being in Africa, that's still a risk. It might be at some point malaria or mosquitoes, which is the vector, can spread in a larger area and might be affecting larger areas. So we have to put more effort in that research as well, and applying vaccines. And cancer immune therapy, it's of course it's a major field, and either targeted through building memory or stimulating immune system, or not targeted, as what we saw today from Dr. Berna. That's very, very interesting to investigate more, because natural killer cells is really something filling the gap between innate and adaptive immunity. So a better understanding for that particularly, it's really important, and will be very useful for immune therapy. And I would like to thank you so much. Tomorrow we'll have a very, very important session. We'll be focused more in hepatitis C vaccines, non-communicable vaccines, which is, I call it the crazy vaccine, non-traditional vaccine. So it'll be Professor Atrabi tomorrow speaking about this. And Dr. Cesar will be speaking about the new mRNA technology in vaccines. And Professor Sir Adrian Hell will be covering everything basically we do in our institute at the Jenner. And after that, there will be a discussion session, so don't miss that. And thank you so much for attending this session.